Okay, we're broadcasting today. We're waiting for some people to join, but my name is Amy Powell. I want to welcome everyone to the PhioPara Tumor Tissue Donation Webinar. We want to talk to you about donating fresh tumor tissue to a really unique and impressive research effort that's going on right now. This research has been funded by 3 different organizations that support research into pheochromocytoma and paraganglioma: Paradifference, the SDHB PhioPara Coalition, and the PhioPara Alliance. And we'd like to thank all 3 of them for this extraordinary effort. Today we want to talk to you about how you can be involved, because this is one research project in my years, all of my years of being involved with this disease. I can't think of anything where patient participation has been more important than it is with this particular research effort. So today we have some panelists who will tell you the what and why and how of your participation and the project in general, so you can understand what it means to the community and how you can get involved. One of the ways that you can get involved, whether or not you have surgery coming up, is to work with us as a patient ambassador. We really need to get the word out, and I can't stress that enough. And it takes the whole community, I think, pushing to get the amount of tumor tissue that we're going to need in the next 18 months. And so if you are interested in being a patient ambassador, you can feel free to contact me, Amy Powell, through the PhioPara project or through the PhioPara Alliance. Also, you can contact Stephanie, the Executive Director of the PhioPara Alliance, and we'd be happy to set you up with information on how you can support this extraordinary effort. So today, like I said, we will be launching into the what, why, and how of this research project, and we're going to start with the what and why. And one of our panelists, Steve Furlong, is here to talk to us about that. I'm going to read his introduction while we're waiting for people to join us today. I'm hoping I'm not jetting through things too quickly, but I want to tell you a little bit about Steve, because he's really important to this project. Steve has over 35 years as both a basic and clinical research scientist. He received his PhD from Michigan State University and did a postdoctoral fellowship in physiology and biophysics at Harvard Medical School. Subsequently, he was on the research staff at Brigham and Women's Hospital and a faculty member at Harvard Medical School. In 1997, Steve joined AstraZeneca in Wilmington, Delaware, and spent the next 18 years in a variety of roles in preclinical and clinical drug discovery and development. Steve has numerous family members who are PPGL patients, which has resulted in his longstanding interest in the search for better drug treatments for this disease. Earlier this year, Steve worked with staff at the Broad Institute of MIT and Harvard to conceive and develop the project we'll discuss today. He will also explain why patient samples are required to drive the project. Steve, welcome, and thank you for joining us today.
Thanks, Amy. So I'm just going to bring the slides up. Hopefully this will work. Can you see the slides?
I can see them, yeah.
Okay, great. So as Amy mentioned, so this is billed as a tumor tissue donation webinar. And so what I'm going to do for the next few minutes is just give you a very brief background for this project that Amy referred to, and tell you why there's such a need for patient samples and how the patient samples will be used. I should say that this is just going to be probably enough to maybe whet your appetite for what's really going to be involved in this project. And so at the end, I've also provided some resources that you can go and you can look at yourself if you choose to sort of dig into the details a bit more. So as you can see on this first slide, we've titled it Partnering with Patients to Create Laboratory Models of Cancer. And that's very much what this is all about. And as I said, is what I'm going to talk about, and then in the next few slides is how the PPGL patient samples will be used for research. And before we go any further, actually, just to make sure that when we refer to PPGL, there's a number of different types of shorter ways that people describe pheochromocytoma and paraganglioma, but that's what PPGL refers to in this context. You probably all knew that, but just wanted to be clear. So in July of this year, there was a press release announcing the beginning of this project, and formally it's referred to as a PPGL tumor dependency map with the three groups that Amy mentioned. And so essentially this PPGL project is piggybacking on a very large effort that's been going on at the Broad Institute at Harvard and MIT for quite some time on common cancers. And basically what this effort does is to map cancer's weak spots and to start to uncover new drug targets for common cancers. So that was the point we entered into the agreement with Broad to apply the technology to study PPGL. And as I'll talk about in the remaining slides, is to drive these studies. It's not only important, it's really essential that we have PPGL patient samples that can be used to sort of inform various aspects of the work. So what is a tumor dependency map? So underlying the concept of a tumor dependency map is the idea that mutations cause cancer cells, or is the primary driver behind cancer cells. But in addition to these mutations allowing cancer cells to grow, it also confers vulnerabilities on the cancer cells that normal cells lack. So the result is that these acquired alterations can represent therapeutic targets. For the majority of cancers, we really don't fully understand the relationship between these genetic alterations of cancer and the dependencies they cause. So the cancer dependency map is basically created to sort of systematically identify these genetic dependencies and the small molecule sensitivities and biomarkers that are tied into all this. So in other words, is that if you understand the underlying genetics, you have the best chance of understanding what drugs might be efficacious for a particular type of cancer. And just to say also that in addition to the common cancers that I mentioned at the outset, like breast and lung cancer and that type of thing, that the Broad has been studying, is that it's not just FEO and PARA or PPGL that is going to be looked at. There are a variety of other rare cancers, but we're kind of at the forefront of this, which to me presents an incredible opportunity for the patients that are that we care about in our group. But the other thing to think about here is that the cancer dependency map is that, so there's a technical aspect to it in terms of understanding the genetics, but the sort of practical aspect to it is that the Broad Institute is highly collaborative, and they always want to share the results of their work. And so the idea is that this cancer dependency map will become a freely available public resource that any researcher anywhere can use to prioritize the most promising drug targets or drug repurposing opportunities for each cancer. And I'll just say briefly, for those of you that aren't aware what drug repurposing is, one of the fastest ways actually to get a new drug to market for a new disease or an indication is to take a drug that's already been approved by the FDA for something other than, in this case, PPGL, because it's already passed through many of the hurdles that slow down, drug studies starting from a totally new concept. So whereas a new drug can take easily a decade to go from defining the new target to drug approval, the thought is that repurposing drugs can go much more quickly than that. So there's three main aspects to these studies: the cellular models, genetic studies, and drug screening. And for each of them, the patient samples are critical. In the case of the cellular models, what will be done is to put them through something that the MIT Broad group refers to as the cell line factory. So the idea is that the PPGL cells that have been developed up till now are suboptimal for doing drug screening. And so the hope is that with the samples that we are collecting, they have a very sort of rigorous approach to developing new cellular models, trying sort of like a matrix of different types of conditions. And they've had a lot of luck with doing that, where other cases that it's been, you know, challenging. So one of our hopes from these studies is that we'll develop new cellular models that will be useful in terms of drug screening. The second aspect to it too ties into the tumor dependency map that I mentioned, and so these are the genetic studies. So probably everybody that's involved in this call is aware that there have been enormous strides made in understanding the genetics of PPGL in the last 2 decades. Members of my family were first diagnosed right around 2000, in that neighborhood. And in those days, there was really not nearly the level of genetic understanding that's available now. But there's still more to be done, and the genetic studies that will be done by the Broad are very extensive and will help us understand gene-gene interactions, for example. The third thing is the drug screening, and so this will be facilitated by the new cellular models, informed by the genetic understanding. And the thing that I didn't mention when I was talking about drug repurposing, for example, is part of the Broad effort has been to collect samples of thousands of drugs that could be screened. So the majority of these drugs are ones that have already been approved for use in humans for a variety of different diseases. And one of the things that they've been finding from their studies with common cancers is that some of the drugs that look like that they may have some efficacy against tumors are ones that were developed for diseases that you wouldn't think would necessarily be tied in. An example being diabetes drugs. So this project is intended to run for 3 years. Our goal is to collect 30 patient samples in the first 18 months of the project, as Barbara will tell you about more in her presentation. And the point, though, that I wanted to emphasize is that the successful collection early in the project will help us have the best chance of accomplishing our aims relating to the cell culture genetic studies and drug screening in the subsequent 18 months. So that if we sort of front load the project with these samples in the remaining 18 months, we can accomplish the other things that we had hoped to do. And then I'll just end on this slide. So the Broad Institute efforts around cancer are amazing, impressive, and an incredible opportunity, I think, for our patient community. If you want to dig into some of the details, which we don't have time to get into today, I've just shown some screenshots on this slide of some of the different online resources that are available. And then below, there are URLs to get to these different things. So as you can see in the upper left-hand corner, there is just an overall description of the Broad Institute, how it was formed, the history, and that type of thing. Below that, there's something that they refer to as the Drug Repurposing Hub. On that site, there is a video describing what drug repurposing is. Next to that, there is a much fuller description of the dependency map than what I gave. And some of it's a little bit dense, but I would encourage any of you that are interested to dig into it. I think it's pretty approachable in terms of providing an understanding of what's involved. Above that is the cell line factory that I mentioned, and just a few details on that. And as I said, the URLs are for each of those are below. So that was it from my section. And so, Amy, back to you.
Okay, thanks, Steve. I'm sure there'll be questions, and I do want to remind everyone that we will be taking questions at the end of Barbara's presentation. Until then, if you have questions, make a note of them, or you can go ahead and submit them, and we will address them. We're going to be devoting about a half hour to questions, and so I encourage you to ask away. No question is too trivial for us to address today. We really want to make sure that the community understands this and has a good understanding of why we're doing it and why your participation is just so vitally important. So to talk about how you can participate and how easy it is, we're going to be talking next with Barbara Van Hare. Barbara is the Director of Foundation Partnerships at the Rare Cancer Research Foundation, which provides infrastructure tools to help further research for low incidence and underfunded cancers. In her role, she works with patients and patient groups to effectively communicate the opportunity to donate tissue for innovative research projects, oversees the patient sample collections, and helps onboard new lab partnerships. She's particularly dedicated to this work as her father, brother, and nephew have been diagnosed with different rare cancers, and she enjoys working with the patient communities across many rare cancer indications. Barbara, thank you for joining us today.
Thank you for having me. See if I can get my screen up here. Can everyone see my screen?
No, not yet.
Can you see it now?
Here we go. Yes.
Okay. Hello, everyone, and thanks for having me here this evening. As Amy mentioned, I'm Barbara at the Rare Cancer Research Foundation, and I'm going to talk to you about why tissue donation is important, and if you're interested in donating tissue, how you can do that fairly easily through a website called pattern.org that is an initiative of the Rare Cancer Research Foundation. So tissue donation, as stated by the NIH, is an immensely important gift to the research process. Medical researchers use tissue donations to make important discoveries that improve the detection, diagnosis, and treatment of cancer, basically as Steve mentioned. And basically, the disease models that are created from these patient tissues help scientists understand the biology of the disease, identify new ways of diagnosis and prevention, and also test and identify new therapies and hopefully cures. As I mentioned, pattern.org is an initiative of the Rare Cancer Research Foundation. And the reason why we launched Pattern was to enable patients such as yourself or others that have rare or low incidence cancers to donate tumor tissue and medical data to high impact research. We basically bridge the gap between patients and scientists, empowering you, the patient, to be a partner in the research process. So basically how Pattern works is if you have an upcoming surgery, you go on to pattern.org and you register with some basic information such as your name, the indication that you've been diagnosed with, and based on that indication, you'll be matched to a particular study. In the case of PPGL patients, they'll be matched to the Broad. And I'll tell you a little more about that project in a minute, but you'll be basically attached to that project where you will see information about the project, and a consent will come up online. You will read through the consent. You can stop and ask questions along the way, either through a chat box or their phone numbers that you can contact. You can pause along the way if you want, or you can go through each page and at the end, go ahead and consent if that's your choice. Basically, you would consent online by signing there on pattern.org. Once you consent, then literally within about 60 seconds, we are notified here at pattern.org/RCRF, letting us know that a patient has consented. We store all of your data in a HIPAA-compliant database. We would go in and see when your upcoming surgery is. If you don't have a date yet, that's okay. If you're having a surgery within 6 months, then you can go ahead and register at Pattern, and we will keep in contact with you if you know more about when your surgery might be. That's really all you have to do at the onset is go to Pattern. If you so choose, consent online, and then we'll take it from there. We'll get in touch with your surgeon as well as any other folks at the hospital where you're being seen, whether that be pathology, operating room staff, whoever there at the hospital that might need to be involved in getting them to release the tissue to us. We will handle all of that for you so that you don't have to. Should you choose to mention it to your doctor, which is great if you want to, but you don't have to, that helps us kind of get the process started, but there is no reason why you have to mention it to your doctor if you don't want to, or if you're uncomfortable with that. We are glad to do all of that work for you. Basically, what we will do after we contact the doctor's office and either through him or others at the hospital, give us an address in which to send a collection kit. We will have a kit overnighted to the hospital. It has to stay at a certain temperature, so once the hospital receives it, they'll refrigerate it, and then we'll work with them, included our instructions, and we'll work with them for any questions they have. It's fairly simple on their part. We include everything in the kit. All literally they need to do is to put the tissue into the sample kit and blood if it's available. If it's not available, that's okay. And then seal it back up. We have a pre-addressed, pre-stamped FedEx label that they would put on the top, and then we arrange for FedEx to pick it up that day. Unless the surgery goes late, then we'll pick it up the next day, and then we will overnight it to the Broad, and it will be there the next morning. Within a couple of weeks of either when you consent or your surgery, we will also send you by mail a medical release form. All of the information is de-identified before it goes to the Broad, but that just allows us to get any history on your disease, such as any therapeutics you've been on, or other information that will help the Broad do their work more effectively. So we will mail that to you with a pre-stamped, pre-addressed envelope. So we try to make it as easy as possible for you. Regarding the Broad project, the tissue will actually go to the part of the Broad called the Cancer Cell Line Project. As I mentioned, all of the tissue in your data is de-identified, so the Broad does not know who you are. The hospital is instructed not to send any identifying information to the hospital. We send with the kit a special ID so that we know where the tissue is from, but the Broad doesn't know the patient specifically. The Broad will then attempt to create cell lines or organoids. The reason why we need fresh tissue directly out of surgery is because they are creating living models. So therefore, we aren't able to use stored tissue for this particular project. They will create models that then they will validate to make sure they are what they are supposed to be. They will sequence them, and then they will also send them to the tumor dependency map part of the project, which Steve described. Also, any models that are created will be shared openly with the worldwide research community. They'll be sent to an ATCC, where it's a public biorepository where researchers worldwide can have access to those models if they want to use them for research in their labs. The importance of this project is, as I mentioned initially, there are potentially issues with researchers not always sharing data as quickly as we might like. Particularly with a rare disease, limited patients may be seen at one hospital. So by working through Pattern and this project, regardless of where you're seen in the continental US, that can be a research institution, academic center, it can be your local hospital, it can be a regional hospital. We will work with the folks at that particular institution to get your tissue sent to the Broad. Some institutions may not have a use for the tissue, in which case some institutions may toss tissue. We're trying to make sure that your tissue is utilized so that we can move the research forward. All of the tissue will be utilized. Once it gets to the Broad, literally they initiate it right away in their lab. Even if there's a surgery on a Friday, the Broad is technically closed on Saturday. In that case, we'll deliver the kit to a FedEx that's nearby the Broad, and one of their staff will go to that FedEx office and pick up the kit, take it over to the Broad, and start initiating it in the lab on Saturday. As I mentioned, these go to the Broad Institute researchers. They're creating cell lines and organoids. and all models and associated data at any part of the project will be placed in the public domain. Many times I'm asked questions on how much tissue you're going to take, what about tissue that's needed for my care. First and foremost, the patient, you all are most important. So the only time we will collect tissue is when it's excess tissue. We state very clearly up front in our conversations with the hospital staff that whatever is needed for your surgery and your care needs to stay there. We will only take tissue that's not needed for your care. The sample size that the bro would like to have is a 1/2 centimeter by 1/2 centimeter. So it's a fairly small piece of tissue. If there's not enough tissue collected from the surgery, then again, whatever's needed for your care comes first. And then we just work with the hospital to re-get that kit so that we can use it on another project or another patient sample. We would ask that you consent prior to your surgery. As I mentioned, we need fresh tissue. And if possible, we know it's not always possible, if you can consent at least a week in advance, that gives us the best chance of collection. If you're unable to give us that much notice, we will do our best to collect with even a day or two. Some exciting news that I have to tell you that literally just happened the past 2 hours is a PPGL patient consented over the weekend. We were able to work with the surgeon yesterday in the hospital where that patient was having their surgery. We overnighted a kit through FedEx yesterday. It arrived at the hospital this morning. The hospital was wonderful to work with. They got the kit to pathology. The surgeon took, after the surgery, a piece of the tissue to pathology to get it done right away. And literally it is in FedEx's hands right now on its way to the Broad. So if you don't have a week in advance to let us know, we'll do our best. We can't guarantee, but we will do our best. Again, we'll mail the medical record institutional release forms to you within several weeks, either after your consent or your surgery. If you're consenting several months in advance, we'll send it sooner. If you're consenting just a few days before your surgery, we'll send it within a couple weeks after the surgery. The project is for general research only, so unfortunately we can't give any of the research results back to the patient. The Broad will release findings from their studies, but it won't be patient specific. And so unfortunately we aren't able to give it back to you. That is a goal that we have for the future and are working with the Broad so that at some point down the road, we hope to be able to do that. But at this present time, we aren't able to. As I mentioned, all the tissue and data is de-identified, and any data associated with any patient is encrypted on a HIPAA-compliant server. So there's very restricted access, so you don't need to worry about us releasing any personal identifying information. If for some reason you were to consent and decided the next day or the next week or next year that for some reason you wanted to revoke the consent, you are able to do that either by going back to pattern.org and signing in, and then we will notify the Broad right away. Any research that had already been done with your tissue, that would have to go forward, but if there was any tissue remaining, that would be destroyed. And there is no cost to you to participate in this research. The results to date, I started working with the 3 groups, literally, I guess it was 2 months ago, 2.5 months ago. And on here, I have an update because this was as of this morning. As of this morning, 2 samples have been delivered to the Broad, but 1 is on its way and should arrive by 10:30 tomorrow morning. So 2 of the patients that did have upcoming surgery, that's down to 1 because 1 had their surgery today. Of the 6 patients that consented, 1 surgery was canceled and 1 sample was uncollectible. But already we have 3 samples and we have 27 more to go. So if there's, if you have any interest in consenting on Pattern and allowing your tissue to be utilized, you can go to pattern.org. Again, if you have any questions, you can use the chat feature on Pattern, or you can contact me directly with questions. My email is on here as well as my phone number. You can feel free to reach out to me. And if you have specific scientific questions, I can get you in touch with the folks at the Broad. Amy, I think we may be ready for.
Okay. So again, if you're joining us late, we're talking about PhenoPara tumor tissue donation in support of the Rare Cancer Dependency Map Project with the Broad Institute. We have Steve Furlong and Barbara Van Heer on the panel here today to answer your questions. I want to talk just a moment as we're waiting for people maybe to think of their questions and submit them, that we do need also patient ambassadors to be involved with this project, even if you don't have any upcoming surgeries. And we hope that that's the case for many people who have been diagnosed and already have their surgeries, that you're stable and disease-free. We still need your help to spread the word about this among the community, in particular on the Facebook support group and on Twitter and Instagram. Because as we all know, newly diagnosed patients turn to online support communities for information, and these are the people that we're going to have trouble reaching, and we could really use your help. So we're going to ask you all to consider becoming patient ambassadors. You can contact myself or Stephanie with the PhenoPara Alliance. I'm Amy with the PhenoPara Project. We'd be happy to share information on what the expectations would be. They're minimal, but we do have some guidelines we'd like to share with you, and then we'd like to put you to work getting the word out with the rest of the community. I don't see any questions submitted yet. I don't know if they're not showing up for me, so I'll ask one that I think some people might have. Just generally, I think that we'll have patients that would wonder, do you want specific gene lines, or are you looking for anyone who has had a pheochromocytoma or paraganglioma?
Yeah, that's a question that's come up before. I mean, it's a good one. And I think the decision is that initially we'll take whatever genetic background we can get. Ultimately is, we would like to have a broad representation of the different types of genes that are known to be relevant. For example, succinate dehydrogenase B is obviously one that's one of the more problematic, mutations. In some cases, patients that are newly diagnosed might not have their genetic information when they get their first surgery. For patients that are coming in maybe for second surgery, the expectation is that they would have an understanding of the genetics. So I think the idea is just initially we're just going to let it go, and hopefully it will just sort out kind of naturally. And if it doesn't, then we might try and do some proactive things to make sure that we get the representation that we want in terms of genetic backgrounds.
Okay.
Does that make sense?
Yeah, it does make sense. Actually, it makes really good sense. Are there any instances, I don't know if either you or Barbara can answer this, where a patient would be looking at upcoming surgery, but because of prior therapies, maybe to attempt to address the tumor without resection or something like that, those have failed and they are looking at surgery again, would you ever say, no, we don't want that tissue because of things that you've gone through already?
Yeah, I mean, again, I think that's there's been some discussion about that, and I'm not sure that there's universal agreement on the answer. But for example, patients who have been treated with MIBG, you could say maybe that would not be the ideal example because presumably you've damaged the tissue with the radiotherapy, but the likelihood is there is still some viable tissue there. So I think the best way to answer that is probably case-by-case basis. So.
Okay. Okay. So everyone should sign up, and then if there's an issue, then perfect. Yep. That's great. So I feel like there was another question that I had there. Let's look and see. We have Leslie, who I think some of you know, she's saying that she's willing to help. We're glad to hear that. And please, like I said, Leslie, please feel free to contact myself or Stephanie and we'll get you going. I don't know, Steve, can you think of any other questions that the community, oh, one question I know will come up, and I know we'd like to address this because we've spoken about it. So many of our patients are being seen at the NIH, and I know many people, actually relatively many, who have upcoming surgeries. Can patients who are being seen at the NIH sign up through pattern.org and donate their tissue?
I could go ahead and answer that. Absolutely. They can sign up with Pattern, and we'll take it from there. We'll coordinate with the Broad and NIH on any cases that are handled there.
Excellent. That's a good, sweet, short answer, but it's the answer that we want to hear.
Amy, I have a couple other points maybe that people have brought up as well that folks might like to hear. Is one of the questions is that, well, if you're not successful right out of the gate developing new cellular models, what are you going to do? And that's something that we actually have a meeting at the Broad October 3rd, specifically to discuss. Art Tischler, who's been working in the field for 40 years, is going to participate and has been very helpful in terms of thinking. Annika Kron, by the way, I need to acknowledge, she's project managing this at the Broad, and she's been fabulous. But I think the point is that there's a number of approaches that we can take if we don't generate the exact cell line that we want, including xenografts, which is a way of placing human tissue into various types of rodent lines and that type of thing. There's ways of immortalizing cell lines that we can discuss. There are new sensitive ways to look at things like cell viability and so forth that may be done just on a couple cells. So it may even be possible to do it on fresh cells. But those are all topics that need to be explored. I would not be the least bit discouraged if we don't have immediate success with developing the cell lines. And we do have other ways to approach it. So,
great. One question that I've also heard from people, because I've contacted people that I know have upcoming surgeries to encourage them to sign up, is I'm afraid there won't be enough tissue for what my doctor wants to do with pathology and then what the Broad Institute needs. So can either of you address that?
I'll address that. Absolutely, whatever is needed for patient care and pathology there at the hospital comes first. So if there's not enough tissue for us to have any, that is fine. That comes first. So
okay, but we'd still encourage everyone to go ahead and sign up because they may be pleasantly surprised at.
Absolutely. And usually the surgeon will let us know up front if they don't think there's going to be enough, but many of them will say, we'll send a kit anyway, just in case. Others will say, we'll need to see after we do the resection. And so we just work with them, whatever is right for that patient. Say if the kit that we send isn't used, we'll reutilize that kit for the next patient. So that's not an issue.
Perfect. Great.
Amy, can I make one other point about drug repurposing? We'll see. Yeah. We're waiting to see if there are any more questions. I think one of the other questions that has come up at various points is, what would lead you to think that looking at drugs that are good for other indications might work for PPGL? And the answer to that is that because there already is some data that suggested that some of the drugs that are in the Broad collection might have some efficacy. And they're pretty early studies, but they're promising enough that I think they'll almost serve like as a positive control that when we start screening things that they're effect. So.
Yeah. Okay, great. So I have another question. This is actually something that I've heard we've had submitted to us. Can I, as a parent, consent for my child who is having surgery? So I would assume maybe that, or even maybe a caregiver who has medical power of attorney. Do you have those instances come up, Barbara?
Yes, we do. So if you have medical power of attorney, absolutely, you can. For a child, yes, there's an extra step that we will walk through once a parent does consent for a child. The Broad has asked that anyone under the age of 21, we have one extra step. And so what happens is after the consent comes in and we see the age, we immediately contact someone at Dana-Farber that is not associated per se with the Broad project, and they will reach out to that parent and the child and walk through with them. One extra step, will email or overnight another consent for the patient and parent to sign for actually assent, and then Barbara will overnight that back to them. We will stay throughout all of that. Is if you are consenting for your child, it's particularly important that you give us at least several days notice so that we can coordinate with Dana-Farber, if at all possible.
Great, great. We've had another issue pop up occasionally. I work with another group, as I mentioned, that has done tumor tissue collection, and they also have said that there are some places that accept postmortem tissue donations. Can you talk to that?
I can. Unfortunately, we are not able to do that. We have done it in a couple of situations in the past, and unfortunately, it gets very complicated. And many times, by the time the patient is deceased, even if they pass in the hospital, by the time the autopsy is done, the tissue has already started degrading. And since the Broad is creating living models, they really need strong viability. So they've asked us not to collect any postmortem tissue.
Perfect. That's a good answer.
If I could just add to that, there's another practical aspect to that too, which is that postmortem tissues can oftentimes have bacterial contamination, for example, and they're reticent to even bring those tissues into their labs for fear that they would contaminate some of their existing cultures. So
that's a good point. Thanks, Steve. So again, fresh tumor tissue, not postmortem, not preserved. We need it immediately post-surgery. That's what we're looking for. I know Stephanie is on the line. I think she's quiet. If anyone has gotten any other questions, now's the time. Again, I see that we have a few participants on the line. I hope that some of you have upcoming surgeries. Now's the time to ask your questions. You will have you won't have as good an opportunity to talk to two experts as you do right now with Barbara and Steve. So if you have any questions regarding tumor tissue donation or this project, now is the time to do it. I do want to ask you too, so we have an 18-month timeline for tumor tissue donation, and then what? What do we expect after the 18 months? What can people look for once we hit that mark? And I know that we're going to get those 30 samples. Then what happens?
well, I think one of the things that I mentioned during my part of the presentation was that there are going to be results from these studies that will be sort of universally available to other researchers. So that's not going to be in the first year, 18 months, just because of the way that these things happen. But as Barbara also mentioned, is that if there are cell lines that are generated, the expectation is that, bits of these cell lines can be given to other researchers to do testing. And all of the drug testing, for example, doesn't necessarily have to be done at the Broad. In principle, the way that they sort of advertise the way that they work is that they can provide parts of their drug library to other researchers to screen and so forth. So it's hard to predict exactly how this is going to go because we don't know what early success we're going to have. An ideal world is that the tissue that we're already brought in is going to start growing, and then we'll be off to the races. But I think that that's maybe a little overly optimistic. So it's probably going to take a better part of the first year and a half just to establish the right culture conditions and so forth. Meanwhile, the genetic studies can be done, and those can be done on aliquots of the tissue samples. As we talked about, is in some cases we'll know the genetic background of the patients in terms of the primary genetic mutation that's relevant to the disease. But even when that's known, the idea would be to try and understand the other gene interactions and so forth, or other mutations that might contribute to the sort of virulence of the disease or whatever. So there will be that kind of information that will come out as well. And then the drug screening part is probably not going to happen until realistically until probably going into the third year. I would love to say that it would happen more quickly than that, and if we get lucky, it could. But again,
will be a lot that can be open. I guess if you're a genetic
publicly shared, but part of the way, so there will be a progress report that will sort of results, presumably they'll have a summary of, the characteristics of the samples that were brought in, whether they're growing or not, whether they have been used for genetic studies. If so, the results of genetic studies. But as I said, it's, that kind of stuff, I think would not be maybe available to the outside for, at least for the first year and a half or so.
Okay.
In fact, I'm probably going out on a limb even to sort of say it's going to happen that quick. So
that's okay. I was still anticipating that for the first year and a half, we're going to probably have some kind of a Christmas tree or a bell or something that we're filling up with, that our 30 donation mark at the top, and hopefully, I just hope it's not an adrenal gland or a tumor that we're deciding to fill in. So, so I think that I know we have at least one attendee online who says that he or she has an upcoming surgery. And again, if surgery is in your future, go ahead and sign up, and everything else will be sort of sorted out by Barbara and her team, and it should be relatively seamless for you. So I encourage, don't second guess it, don't worry about being whether you qualify. Let Barbara sort that part out and sign up. Pattern.org couldn't be easier. I do also want to stress again that we are, in addition to looking for people to sign up and give us tumor tissue, we're looking for patient ambassadors to get the word out to other patients. And another way that people can help, if you have donated tissue or to any project at any point, or if you're considering it, we would love to hear your story, particularly a video story. Some of you may know that the PhioPara project, usually in conjunction with some of the other PhioPara support groups, does a Net Cancer Day video every year. And we would love to highlight this effort for Net Cancer Day this year and have a video story or two about you and your reasons for deciding to donate your tissue, your hopes and aspirations for the research project, and what you're looking forward to in your future as a PhioPara patient. So I will give it a couple more minutes, maybe just to see if there are any lingering questions. I think we've covered the consent issues pretty thoroughly, and I'm glad to hear that medical power of attorney can work. Unfortunately, as we all know, sometimes our loved ones wind up being unable to consent to those things at some point, and it's good to know that, especially if we know their wishes, that we can do so for them. So, and as someone personally who could be in that position, I'm glad to know that. So, okay,
so
I've got
while you're waiting to see if there are any final questions, maybe just like one additional build, I think, on the question that you asked earlier about sort of genetic background. So I think the goal on this is to sort of not only understand, or develop this cancer dependency map, but the ultimate goal is to do what's called precision or personalized medicine. So in other words, is the expectation might be that, there are some drugs that are optimal for some patients based on the genetic background that may not be optimal for patients with a different genetic background. And so that's all part and parcel of the project. And also relevant to the idea of whether patients know their genetic background or not. In some cases, I mean, there's a certain percentage of the patients that, refer to as, a sporadic disease or whatever, so that, you really don't understand what the genetic background is. It's possible that there is genetic information to be had through the work that the MIT group will do, the Broad group, that would maybe illuminate, what that, because, the expectation is that in most cases there is some genetic explanation for why the tumors are developing. And I think that as we said earlier in the presentation, 20 years ago there was almost nothing known about the genetics, and now there's an enormous amount known, but there's still more to be learned. So.
Yep, and we've learned that it's tremendously important to this disease in particular, and that's a really good point, Steve. I have a request that I could, if asking me to elaborate a little bit more on becoming a patient ambassador. So if you'd like to be a patient ambassador, contact myself, Amy Powell. You can contact me through Facebook. I'm easy to find. TheoPara Project is on Facebook. You can contact us there. Or you can contact Stephanie at the TheoPara Alliance, and we will get you set up. It's a pretty easy process. We just ask that you post occasionally to social media, either directly about this project or post comments on other people's posts about the project, just to keep awareness up. I think, as many of us know, our networks are different. Sometimes they overlap, but you probably reach people that I don't reach, that Steve doesn't reach, that Stephanie and the Alliance even don't reach. So we really need those people to, be our 6 degrees of separation, because there's, all of those patients out there that we're peripherally connected to, and you're very important to getting the information directly to them, because otherwise they just may not hear it. So we do have some basic guidelines that do sort of lay out, again, what I was just saying about the occasional posts, and I think generally anything is fine, but we do want to make sure that we set expectations. So we have some guidelines for social media posting that we would probably hand out for people to ask that you would adhere to that. And it's very easy, and I think that you will be surprised pleasantly at how easy it is to support this project in a really meaningful way. So I hope, Jennifer, that answers your question, and if not, please email me. You can, again, contact me through Facebook or contact Stephanie. She's info@theopara.org. Also, the TheoPara Alliance is on Facebook, and the TheoPara Project is as well. And I know that we all are eager to connect with you and get you up to speed and on the streets, so to speak. So I think that may be the end of our questions. I'm not seeing anything in the Q&A spot, and I don't see anything else queued up in our chat. So I do want to thank everyone who took the time to attend today. I also want to let everyone that's attending know that this will be recorded. So if you know someone who is kicking themselves because they missed it or they were working and didn't have time to join in, this has been recorded. It will be online and available very soon. And again, you can contact myself, Stephanie with the TheoPara Alliance, and Barbara with her organization, or Steve, and any of us can answer your questions. And really, I think we get you where you need to be on whether you are donating, whether you're going to support this as an ambassador, whether you're going to submit a video or a written story to us talking about your personal tissue donation story. So we're tremendously excited about this on behalf of the Paradifference Organization, SDHB, TheoPara Coalition, the TheoPara Alliance, and the TheoPara Project. I want to thank everybody for attending, and in advance for the hard work you're going to do in supporting this really important research project. Thank you.